Okay, welcome everyone. This is part of the program, but it's also the ICTS monthly colloquium. We like to have the colloquium as an opportunity for members at ICTS to also get to know what programs are happening. And one of the best ways to do that is to have speakers from the program actually give our monthly colloquium. The talk will have in-person participants from the ICTS community as well, plus people online. Usual rules apply. If you have good questions, please do wait for the microphone so that online participants can also hear your question. Other than that, I don't have much house rules to say. I'll hand it over to Ganga, who will introduce the speaker.
Thanks, Vishal. Yeah, we're happy to have Joel Marthelot. He's also one of the co-organizers of this school. So I've known Joel for a very long time. So Joel did undergraduate and PhD from ESPCI in Paris, and then he was at MIT and Princeton for a postdoc, and then after that he's gone back to France. He's now a faculty, CNRS faculty at Aix-Marseille University, IUSTA. Over to you, Joel.
Thank you, Ganga. Thank you for coming. So I'll talk about morphing without muscles. And this is motivated by, we'll see a couple of examples of hydraulic actuation in insects on plants. And so I'll motivate a little bit this with an engineering perspective. So there is a growing interest in engineering community to go from rigid to soft actuation. So, okay, we know very well how to do rigid robots, and there is a growing interest in building softer robotic solutions. And associated with this shift, there are challenges to program the target shape, the dynamics, and the mechanical response of such soft actuators. And it turns out that there are many examples in nature, biological strategies, where we see this kind of very large transformation of shapes in substructures. And one is the deployment of the wing of insects when they go out of their pupal case. And so I'll discuss this in more detail. And so the system I'm interested in and I'll present today is are systems where the motor is very simple, is a change in pressure, but it can play on a structure which is anisotropic or organized in network, such that the motion is complex. And so there is also this underlying question, which is how to try to program this motion with the anisotropicity of the structure. Okay, and so in the team, we are interested in both more engineering perspective, so bio-inspired actuation. So I'll not discuss this today, but I'll focus on several examples of morphing without muscles in nature, starting with insects, and then moving to plants, and maybe, if I have time, discussing a little bit eversion mechanisms. Okay, so feel free to ask questions anytime. So I'll start with wing expansion in insects. So we are focusing on Drosophila melanogaster. This is the work of PhD student Simon Judge, who recently defended. And so this is an example on cicadas. So this is a cicada which just going out of its pupal case. So cicada, very common insect in Marseille. And this is a sped up movie. What you can see is there is a tremendous change of shape in the substructure, on a fast expansion of the structure. This is a paper, this video is from a paper by Marie Salcedo et al. And so the video was sped up. The total time it takes for the insect to deploy its wing is about 20 minutes. But this is quite original in animal morphogenesis, because this is a change of shape at the level of the organ, which occurs in minutes. And it's a very large change of shape. So the total deformation of the structure is about 400% in wingspan. And it's not associated with cellular processes. So there is no growth or contractility of the tissue associated with transformation. On something I'll not discuss today, but which is also a source of interest, is that this transformation is behavior dependent. So if the insect feels threatened, it will not deploy its wing. So as I said, in the lab, so we originally tried to work a little bit on cicadas, but it's not so easy to find cicadas going out of their pupal case. And so we turn to Drosophila, which is the usual biological system, one of the most used biological systems for development. And so if we are looking at what is happening for the Drosophila at the when it just get out of the pupal case, what we can see is that essentially what the Drosophila is doing is swallowing air and contracting its muscle, abdominal muscles. And the digestive tract of the fly is a closed system, while the blood system, so hemolymph, which is the technical term for blood in insects, is an open system, which means that the whole body of the insect baths in his hemolymph. So the basic idea is that because of this swallowing of air, the digestive tract volume will increase like a balloon, but it's surrounded by hemolymph, which is this open circulatory system, such that the hemolymph pressure will increase. And this increase, if it's large enough, will drive the blood inside the structure, which is the wing. So this is a typical observation of the wing deployment. So what we can see is that we see this very large transformation of shape. I will play the movie again. If you focus on this side, what you can see is that we have a transformation where we have a lot of different curvature occurring during the transformation, but at the end of the process, the wing is completely flat. And if you now focus maybe on the head of the insect, what you can see is that the head is quite expanded now. And at the end of the process, we'll see that the head will completely deflate. Okay, so we see that this increase of pressure is actually occurring in the whole body of the insect. Okay, and at the end, the process is irreversible, so the wing is completely transformed and will stay like this, flat, during the whole life of the insect. It's a critical moment for the insect. If it fails to open its wing at that time, it will essentially be unable to fly. Okay, so we can start by trying to look at what is the kinematics of this transformation. And so to do this, what we can do is to do microtomography of the wings. So what we see is that we have these very big folds. Okay, so these macroscopic folds, which look a little bit like an origami. Okay, but if we now focus on one of these cross-section, what we see is that, in fact, so these macroscopic folds, they are composed of two layers, okay, which are separated by almost a constant thickness. Okay, so two plates essentially separated by a constant thickness, and they are linked, these two plates, by some kind of pillars which are connecting one plate to another. Okay, and if we take a cross-section of this view, what we see is that these pillars, they are actually organized in some kind of lattice of hexagonal organization connecting these two plates. If we now try to understand what is the flow of hemolymph in the wing, what we can do is we can inject some microbeads fluorescent in the abdomen of the insect before deployment. And what we see is that these are some kind of tracers of the flow. We see that the hemolymph is actually going in the whole the wing during deployment. This is very different from what we would expect looking at what is happening for the flow in an adult wing. So in an adult wing, whole the process is happening in the veins, while during deployment, there is no specific things happening in the veins. The whole empty space between the two plates essentially are invaded by the blood. So this is very similar, in fact, to structure that we are familiar with made by like inflatable matrices or structure like this. So a very first naive picture of this structure is essentially we have two plates which are quite thick, and they are connected by some kind of pillars that are organized in an hexagonal lattice. On the next question we want to ask is, what is the transformation? Is the transformation inextensible? Like if I take a sheet of paper, an origami sheet, and I'm stretching on it, I will get an inextensible deformation. And we are lucky to have tracers, in fact, of the transformation of the length in the wing, which are the position of the veins that we can find in the folded state and compare in the expanded state. And so this is the reconstruction of the 3D reconstruction of the sum of the veins that we can track in the folded state. And we can compare the arc length of each of these veins in the folded and deployed state. So this is at scale. And what we see is that the deployed length is, in fact, 60% larger than the folded one. Meaning that not only we have this macroscopic fold, like in an origami that essentially unfold, we also have an extension of the wing, which seems to be actually the same in all directions. So if you look at veins in this direction or in this direction, we see an expansion which seems to be 60% more or less in all the veins. So isotropic in the plane. So if we want to understand at smaller scale what is happening, what we can do is to take cross-section of the wing and to do some TM, electron microscopy. And what we see is that we see again these two plates, which are in fact made of monocellular layers of epithelial cells. But they are in fact, we find back the pillars which are connecting these two monocellular layers. But we also see that, in fact, there is a rigid thin layer of cuticle, which is much stiffer than the epithelial cells, which is initially completely wrinkled. Okay? And if we look at characterization, biological characterization, what we can see is that in the pillars, there are structures like microtubules and actin that are connected at the basal junction of the two monocellular layer of cells by interlocking integrin structures. Okay, so we have this kind of picture in which we have two epithelial cells, which are depicted in green, pillars, which are reinforced by this kind of actin fibers, on initially wrinkled cuticular layer. Then we can try to see what is happening during deployment. So this is not an easy task because the wing is actually changing a lot its curvature as we've seen and so on. But we can do still two photon microscopy in order to try to see what is the typical scale between these two cellular layers during deployment. And what we see is that during deployment, the pillars, they tend to stretch, essentially. So we see that the distance between the two cellular layers tends to increase under pressure due to the increase of the pressure of blood inside the structure. So the basic schematic would be that we are now pressurizing the blood in the cell, and this will lead to some variation of the length of the pillars and a variation of an extension of the cellular layers. And what we see in the deployed case, so back to electron microscopy, is that at the end of the process, the cuticular layer, which was initially wrinkled, is now completely flat. completely flattened. So there is also another thing, which is that we don't see the cellular layers anymore. And so this is something which is happening later in the process after deployment, which is the fact that there is an apoptosis of this cellular layer, which is then sucked out of the wing. So this is a process that was characterized a little bit biologically speaking, but not too much in terms of mechanics yet. At the end of the process, the cells are completely washed out of the wing. Okay, I'll not discuss this because this is occurring after deployment. So at the end of the process, what we see is that the wrinkles, the initial wrinkles, they have completely flattened. So we can try to characterize more quantitatively this kinematic at the small scale. And the first thing we can do is to do profilometry of the structure. So we have an initial folded structure in which what we see is that we have this hexagonal lattice organization, which corresponds to the hexagonal lattice organization of the cell. And we have some wrinkles initially with a typical amplitude of about 4 micrometers on a typical scale of 7 micrometers. And after deployment, what we see is that everything is much flatter. So the amplitude is now about 1 micrometer. On the size of the cells, they have increased quite a bit. So we get something like 13 micrometers. So we can record this change of surface area of the structure, and also record what is the actual 3D area here, because it's very flat, corresponds to the surface of essentially very close to the surface area. And because we are playing with Drosophila, we can mark different things in the cells, and one thing we can mark is the cell contour with cadherin mutants. So we can track the variation of the surface of cells before in the folded and in the deployed state. What we see is that the area of the cells, they tend to increase during the pressurization, meaning that the cells, they are stretched. Okay, so this cellular layer is actually stretched during the process. But we can also mark the apical surface of the wing with another mutant, utrophin. And what we see is that above each of any individual cells, you get a structure like this, which is some kind of conical structure, where you have, so this is the wrinkles that I was discussing earlier. So in fact, it has a three-dimensional shape, which is very well conserved from one cell to another. And if we are looking at what is the 3D surface area of this pyramid-like structure, we see that there is enough surface to map the deployed state, meaning that you don't need to stretch this thin film layer, which is on top of the structure. And if we sketch the process, we see that, in fact, we have a two-scale deployment mechanism in which we get this large macroscopic unfolding of the big folds, but also expansion at a smaller scale, which corresponds to, with this simple schematic bilayer system, an increase of the length, so stretching of the epithelial cells on a non-wrinkling of the thin film layer. Okay? So this is a little bit the picture in terms of kinematics. And then, yes, go ahead.
Which one happens first?
Yes, so I think everything is a little bit coupled. I would say that still the unfolding process, the macroscopic unfolding process tends to occur first. And at the end of the process, you really have extension of the tissue. But this is very schematic. I mean, everything is slightly coupled, in fact.
Yeah. Another question, I think it was in the previous slide.
Yeah.
Before this.
Before this one.
Yeah. The third picture. next picture on the right. No, this one. Yeah. So here, what happens from the middle to the right one?
From this one to this one?
Middle, yes, yes.
Yes, so essentially what is happening is you have completely stretched the cuticle. Sorry, you have completely unwrinkled the cuticle without stretching it, okay? On the cellular layers, they die and they are washed out of the wing, okay? So this is a process which occurs after deployment.
Okay, we just collapse.
Yeah, exactly. So if you look at the thickness of the cuticle at the end of the process, it's exactly the same as the one at the beginning. So really, that thing completely unwrinkled without being stretched. Okay? Other questions? Okay. So then we can try to measure what is the pressure that the pressure of the blood during the deployment. So to do this, what we do is we connect the fly, which is very small, to a pressure sensor. So there is a homemade setup in order to measure this very small, the pressure in this very small insect. And so what you do is essentially you're puncturing with a glass tube the abdomen of the fly. On what we see, if we look at what is the variation of the pressure in time, is that the pressure tends to increase and reach a plateau, and then deployment occurs, and then there are irreversible processes occurring, and the pressure in the fly decreases back to its normal pressure. Okay, so we get this increase of pressure of a few kilopascals, and then we can do very simple mechanical argument, just looking at what is the effect of pressure on this structure. So what we do is essentially we are looking at what is the effect on the pressure with respect to the stress which is applied in the big plates. And the only thing we need to be careful about is that because the variation of the geometry varies quite a bit, there is a variation in the thickness, which is thin during the process, and in the surface on which the pressure is applied, which increases. Okay? And we assume an incompressibility of the cellular layers in this case. And we can do exactly the same force balance in the pillars. And so we get the same idea that when the pressure is applied, basically, it tends to stretch the pillars and also decrease their diameter. And we need to take into account the variation of geometry. And in order to predict what is happening, we need a constitutive law for the wing. And so we started to try to measure what is the force displacement relation on this wing structure. And so what you can do on a dead fly is to stretch the wing and measure the force. So what you see, if you look at what is the stress as a function of the displacement of the sensor, is initially you have a very nonlinear response because you are essentially unfolding the big macroscopic folds and then started to stretch the tissue. So if you want to get an idea of what is the constitutive law of the material, you need to look at what is the variation of the actual length in the structure. So we plot now the stress as a function of the stretch. And what we see is still nonlinear, but slightly less. And we can put on this curve a very crude phenomenological model. So what we see is that it tends to strength stiffen. Okay, and so we can interpret this physically as the fact that initially what we do, we are essentially stretching the cellular layer, which is much softer. And at some point, you start to stretch the epithelial, the cuticular layer, which is much stiffer. And so you start to have a large increase of the stiffness of your structure. And so if you put now this strain stiffening, so we can take a constitutive equation with just 2 parameters to try to capture this trend stiffening behavior with the variation of the stress as a function of the stretch with our small mechanical model. We can predict what is the variation of the stretch as a function of pressure. So what you see is that here I've plotted the stretch of the pillars in brown in the plane in black. Sorry. And what we see is that initially the pillars, they tend to stretch, but because of this trend stiffening behavior, the stretch will saturate. Okay, and when this stretch of the pillars start to saturate, we see that there is a very nonlinear increase of the stretch in the plate that expand in a very small region of pressure. Okay, so now if I'm increasing the pressure, essentially the plate will stay with a small stretch for a given range of pressure and then quickly expand. So this you can also you can just solve the equation I've shown, and you can also do a numerical simulation in order to look at colors. And so you'll see essentially the same thing, that you have this strong expansion of the pillars that saturate, and then a large expansion of the plate. And for the insect, if we focus on the plate expansion, this is quite interesting because it means that there is a range of pressure for which you get an operating point. So a very large variation of the stretch in the plane. And so we can try to test this model by artificially increasing pressure in dead flies. And so this is the case here. So here what we are doing is we're increasing pressure by just injecting water, in this case in the abdomen of the insect. And so between 0 and 8 kilopascal, what we see is that there is no deployment occurring. But now, when we start to increase the pressure around 15 kilopascal, what you'll see is that the wing will indeed deploy. And so the deployment process is not exactly the same as in the living structure. You see that you get some curvature. But still, you get this deployment process around this operating point. And if you increase even more the pressure, you will start to see a different situation in which essentially you are tearing apart the pillars. And so you can see that now you have a structure which is completely inextensible. So it looks like a zeppelin, more or less, in which this structure can't stretch anymore, so you go towards this kind of party balloon shape of the wing. Okay, so what we've understood with this simple model is that there is an operating point to trigger the deployment, and that it's compatible with artificial pressure increase. Still, so the fact that we have this operating point is really something that we see in the normal behavior during deployment. So all the deployment occurs on this pressure plateau, but the exact number of pressure don't completely match. So we have a 5-time ratio between the artificial deployment on the actual one. And this may be due to a change in wing mechanical properties between the dead and the live fly, or also active processes. So right now we are performing laser ablation experiments to see what is the active stresses in the cellular layers in order to see if this can change a little bit the effective stiffness of the wing. Okay? But so everything I've discussed so far is completely steady. And what you can see is that, in fact, there is some kind of time scale which is hidden in this process, which is that at a constant pressure plateau, we still need about 5 to 10 minutes to completely deploy the wings. Okay, so now I'll try to discuss a little bit this what expand because of the pressurization of the structure, and that we can capture the mechanical and dynamical properties of the structure by coupling this viscoelastic constitutive law with the geometry, which gives you this operating point on the typical dynamic of opening of the wing. Okay? So I'll switch to another part afterwards, so feel free to ask questions if you have any. Well, I can, yeah, go ahead.
Can you say a bit about the experiment where you inflated externally and the wing became saddle-shaped? Do you have a sense for why?
Yes, so this is still an open question. So it's funny because developmental biologists, they every time they see this movie, they connect to a mutant they know very well, which is called curly wings phenotype. So we don't know yet exactly what could be the underlying mechanism. The most probable one, I would say, is that if there is a slight variation of stiffness due to drying process during the fact that the fly is dead, you can have this drying process, which is not completely the same in the upper and lower part of the wing, and this will, if you now increase the pressure, will tend to make the wing curve. This is the most probable, but also maybe less exciting explanation I can give you. The thing is, we want to work on these curly mutants in order to understand, because this is not well understood. If this, in curly mutants, it's a difference of stiffness between the top and bottom part, or if it's a difference of length. It's an open question for now in this mutant. For sure, in our case, there is no difference of length. but it's still an open, or if there are active processes, it's unlikely, but you could imagine also there are active processes that regulate the curvature of the wing during the process. Good. Okay, so I'll switch to another system, which is a sensitive plant, which is called Mimosa pudica. This is the work of a postdoc, Mathieu Rivière, in collaboration with Joel Marthelot in the lab. And so Mimosa pudica is a plant which is also known as a touch me not plant. And so what is happening here is that we are bringing close to the Mimosa pudica lighter. So we are increasing locally the heat here. And what you see is that the leaves, they tend to close, and the signal is propagating. Okay, and so this is real time. Okay, so that's the weird thing when you are interacting with this plant, is that the motion is quite fast. Okay, and so why is this plant moving? It has a motor organ, which is called the pulvinus, which is some kind of small rod, and you have a primary, secondary, on third pulvinus. And so we are focusing on this one, the biggest one, which is there. So it's a thick cylindrical rod. The radius is about 0.5 millimeter, the length 3 millimeter. And if we zoom in the process, so another way is not to burn the plant, but just to gently touch it. And so you can see in this movie that essentially if you touch the plant, it will bend. And so this is really like a rod which is bending, a thick rod which is bending. And if you take a cross-section of the pulvinus, what you can see is that it's a fairly cylindrical tube with many cells, in fact, inside. Okay. And so as the people who've followed the class of Adrien just before know, is that the plant cells are very different from animal cells because they can withstand pressure. Okay, and so you can apply a pressure in the cell, and the pressure will be taken by the mechanical stress in the wall, and you can play with turgor pressure in order to regulate what is the volume of water which is inside the plant. Okay, and so the way of seeing this is that this exchange of liquid essentially is the internal muscles for the plant movement. So we can schematize this as a change of turgor pressure will leads to a water flow, and this will lead to motion in the structure. Okay, so this is the picture at the cell scale, but now we are seeing a motion which is at the organ scale. And so the question is, how can we get this kind of fast motion in this specific plant, while most of the motion in plants tends to occurs at a much slower pace. And so if you look at textbooks, it seems that everything is understood. So it's actually a system that is described in textbooks. And people say that it's associated with a fast drop of turgor pressure and a rapid osmotic flow. So the idea is that you have ion flux between one part to the other part of the pulvinus. This corresponds to a turgor pressure drop, and so water will flow from the lower to the upper pulvinus and leads to bending. But something which is not so obvious is that you still need to cross many of these cell walls in order to move the liquid from one side to another. So about 50 cells. And so it's unclear how you can get a process which is that fast. And in fact, if you're looking a little bit in literature, even if it's in textbooks, people refer to papers. So for example, a recent paper, a beautiful paper by Aguirre and Toyota, refer to a paper by Allen in '69. But then if you look back on, everything goes back, in fact, to a paper by Brücke in 1848 that perfectly showed that movements are caused by unequal change of expansion force in the tissues. But so, of course, German scientists in the 19th did the best experiment, as we know, but still we can try to see with new tools if we can understand better this flow. So first question we're asking is, can we draw analogy and differences between animal muscles that we know? So they are driven by acting myosin molecular motors. And actually, this microscopic description is derived by observation at the macroscopic scale by Hill, which is known as Hill's law, which is by looking at experiment in frogs, muscle of frogs. He realized that the velocity of shortening of the muscles depend on the load that you're applying. So the faster you try to move, the smaller the load you can get. And so, can we make any connection with this kind of osmotic motor that we are now seeing? And can we try to understand what is the variation of the force? So here it's, in fact, a torque as a function of the speed, so the angular speed in our case. So to do this, we need to characterize the speed of the motion. So we do this by doing experiments on free motion of the pulvinus. So in order to not, so you've seen that initially what we did is we mechanically triggered the plant, but everything is moving when you do this. So in order to do something a little bit more clean, what you can do is you can apply a difference of potential between one side on the other side of the pulvinus. And in this case, the plant will react to this difference of potential. And so if you put the difference of potential, what you can see is you have a very clean motion of the pulvinus, and you can record what is the variation of the angle as a function of time. And so you see this kind of exponential relaxation with a typical time scale of about 0.6 seconds. Okay, so this is a characterization of the speed. And we also need to try to understand what is the force. So to do this, what we do is we block the motion with here a rigid obstacle, and we measure what is the force which is applied by the plant on the obstacle. And you get the external torque as a function of time, and you see again a relaxation with a time scale which is slightly smaller, but still on the order of 0.5 seconds. And so if you want to understand these two relaxation time, essentially you can build a very simple model in which you only need to have two stiffness, one in parallel to your motor and one in series. Okay? And with this free motion and the blocked motion, you can get what is a variation of the internal torque as a function of the speed by looking at the simple Venus, in looking at free motion on blocked motion, on getting what is the internal torque as a function of velocity. And what you see is very different from what we've seen in animals. So in animals, we had something which had this kind of shape, and here we see something which is quite linear, except at very small time scale. We see that the variation of the internal torque is linear with the velocity. And if we integrate what is the area under this curve, we get a typical power, which is developed by our osmotic muscle. And if we are now looking at what is this power as a function of mass, we get something which is, in fact, very similar to what you see by the power developed by muscles. And so we can try to build a very naive model based on what we've seen just before, the osmotic, so based on a microscopic ideal osmotic motor. So we've seen that associated with variation of turgor pressure. So if I have a flux of ion, I will have a flux of water, which will be correlated with the hydraulic conductivity of the membrane of the cells on the difference between the turgor pressure and the actual pressure in the cell. That we have a real linear relationship between the pressure and the flux. Of course, this is at the level of the cells, and now we can try to look at a simple model in which we are considering that the pulvinus is composed of two reservoirs that will change their turgor pressure, one decreasing and the other one increasing. And we can map, just by doing simple geometry, what is the torque, how the maximal torque is related with the variation of turgor pressure. So just with the geometry of the beam, essentially, on how the angular velocity varies as a function of the hydraulic conductivity of the membrane and the variation of turgor pressure. And so by looking at gamma max and omega max, essentially, we can get a measure of what is the variation of turgor pressure during the process and what is the typical hydraulic conductivity at the level of the organ. And as we know that the number of cells that the water has to cross is about 50, we can go back to what should be the hydraulic conductivity at the level of one cell. And what we see is that there is okay, so we indeed recover something which is linear in terms of torque versus speed, but numbers don't completely match what we expect, because what we've measured is that the hydraulic conductivity of the membrane, deduced from the dynamics on this simple model, is much larger than the reported values for plant cells, 2 orders of magnitude larger. Essentially, the hydraulic conductivity of the cells should be much larger than the ones that are usually reported. On another experiment tells us also that there is a timescale mismatch in this problem, which is if you are now cutting the pulvinus and you're putting it in a bath with a lot of sorbitol, essentially you will dry the pulvinus, okay? And now you take this part of the pulvinus and you put it in pure water. What you will see is that the pulvinus will expand, but if you look at the movie, which is here sped up dramatically, you see that the variation of the length as function of time is of the order of 10 minutes. Okay, so much larger than the time scale of the motion. Okay? So there is an open question, which is how does water can move so fast in a dense plant tissue? And this is related to the motion that we're seeing. Why is this motion so fast? And is the water even moving at all, in fact, in this problem? And so we went back to the movie that I've shown you before, and we had a closer look, and maybe you can see something which is occurring during the motion. Does anyone see something weird? So we see some kind of darkening front, a change of color, okay, during the motion. And if you're not convinced, I can block the pulvinus and trigger the motion electrically and look at with a high-speed camera. And what we see is again this variation of intensity of color in the pulvinus. Okay, so the pulvinus is darkening. And if you are still not convinced, I can look at image difference, and you can see the variation of the front by applying a little bit of a non-rigid transform, because the pulvinus is still moving a little bit. Okay, and so we see that there is indeed a front which is associated with this darkening front with a typical speed of 1 millimeter per second. And so the question we're asking is this an evidence of the fast water transport? And so a way of interpreting this is to go back to the beach. So it's fairly easy in Marseille. And what you see in the beach when there is a wave is that while dry sand seems to be very bright, wet sand seems much darker. And the reason of this is just because essentially when you are sending light on these grains, they have a lot of air pockets in between the sand grains, and so there is a lot of backscattering on low transmission of the light. Okay, but as soon as you have a wave, this pocket of air will disappear, and so you will have enhanced transmission and reduced backscattering, and so it will appear darker. Okay, and so making connection with this, we can look back at the transversal cut of the pulvinus, and we can see that the cells, in fact, they are filled with gas, and so there's a question which is does the water in the cells flow in this gas pocket, which are this intercellular space? And in fact, this is also something you can see in biology, for example, in these skeleton flowers that you find in Japan, in which, again, you get these very white color-like petals, which are due to this intercellular space that completely appears to be darker when when you put some water on it. Okay, so this I'll skip on. So I'll discuss to finish. I'll discuss an alternative hydraulic mechanism in which what we need to show is that there is these intercellular channels, which are initially filled with gas in the pulvinus, and that are interconnected. And what we want to show is also that when we trigger the plant, these air channels there are filled with the water which are expelled from the cells. And at last, we want to understand how this intercellular channels network enable this fast long-distance water transport from the lower to the upper pulvinus. And so to do this, we went to synchrotron, which is based in Paris, Soleil synchrotron. And so you need to build a small setup in order for the plant to be not too unhappy under X-ray, but you can make that. And so you can, thanks to the very hard X-rays that you can get a fast imaging of the inner part of the pulvinus. And so this is a typical reconstruction that you can see. And so if I'm thresholding now the position of the air bubbles, what we can see is that, in fact, this pulvinus structure is extremely porous. So we get these pores, which are interconnected in 3D, and the intercellular space is completely connected in this pulvinus structure. And if you want to characterize what is happening at a smaller scale, what you can do is to do some electron microscopy, and you can find that this intercellular channel are up to the epidermis border. So this is the upper epidermis border of the pulvinus. And you see that you have this kind of small pores, which are submicronic here, that are making this kind of channel network around each of the cells. And so in order to characterize better the geometry of the structure, we did some serial block phase electron microscopy in order to get this kind of typical reconstruction of the channel network. And what you see is that you have a typical channel diameter, which is quite constant if you look at this part of the structure, which is about 2.5 micrometer. So around cellular structure, you will see this kind of interconnected porous structure. Okay, so we can now look at what is happening with this channel before and after you have triggered the structure. And what you see is that initially you have a very porous structure, and after motion, you have a decrease of the porosity of the structure. And so typically you can try to get what is the 3D variation of this porosity, and you get a variation of the order of 7 to 10%. And then you can ask if there is indeed a variation of volume associated with this motion. And so to do this, you can compute the rest on triggered 3D volume of the upper and lower part of the pulvinus. And what you see is that the variation of volume in the lower half indeed decreases linearly with the curvature, while the upper part increases linearly. So you really have a transfer of volume from the lower half to the upper part of the pulvinus. On this, you can also try to do on time-resolved observation. So what is the variation of the volume in time while the curvature change? And indeed, again, you find the same kind of variation of volume in which you are moving the liquid from the lower to the upper half of the pulvinus. So how can we understand this fast transport now of the liquid in the organ? So we've seen that osmotic gradient does not give this fast variation of the flow. So there is a slow cell-to-cell osmotic pathway, essentially. And this is associated with a a timescale which is way too long compared to the one we've seen in the process. But there is another way of moving water in this structure, which is by playing with pressure gradients. And so the idea now is that you don't have the motor is not only the osmotic transfer of water from one cell to another, but due to a difference of pressure in your interconnected channel of pores. And so in order to test this, what you can do is to cut one part of the pulvinus and to let it dry here for 20 minutes, such that you have a slightly dried, you increase the pressure gradient in the pulvinus. So this is the case here. And now when I'm washing the pulvinus with water, what you can see is that in real time, you get this very fast motion of the pulvinus. And maybe you can also see that there are some bubbles here that are expelled. Okay, so gas is really moving away in the structure. Okay, so you can quantify. So this is the case where we've cut the pulvinus. Actually, on what you see is the front propagation here of the motion, which is also associated with a variation of color. On the velocity that we get by building this pressure gradient in the pulvinus is very similar to what we've reported previously. And so the idea is that, in fact, we have a composite pathway in which we have this fast pressure driven pathway in parallel to the slow cell-to-cell osmotic pathway. And you can see by building a very simple model that there is a length scale for which it starts to be actually faster to rely on this difference of pressure rather than relying on the usual osmotic timescale. Okay, so if the organ is large enough, it's better essentially to rely on this pressure gradient pathway. Okay, so with this, we can look back at what is the typical timescale that plants usually lived with, essentially. So there is this usual way of looking at what is the timescale of motion as a function of the tissue size in plants. On most of the plants lives with water-driven motion. And you have a poroelastic timescale that limits the motion at which you can move. And there is a lot of plants that people have looked carefully, like carnivorous plant, for example, that use other ways, like mechanical instabilities, in order to move faster. But here, what we've seen is that Mimosa pudica, they live a little bit below the poroelastic timescale. If you build the poroelastic timescale on the cell size. On this mechanism of playing with this composite pathway of local osmotic flow on long distance pressure driven flow essentially makes the motion faster than usual, such that you are playing with the typical pressure than plants are used to develop. But the interesting part in the case of Mimosa is that you are able to move the fluid much faster, such that you can develop a power density which is much larger than we usually see in other actuation in plants. Okay, so with this, I will stop there. And I will just I'm done, no, Ganga? Yes, okay. I will just acknowledge the fact that the wing deployment story is mostly the work of Simon Adjadj, a PhD student who recently defended on the postdoc in Andrade Silva. On the hydraulic network story is led by a postdoc Mathieu Rivière on collaboration with Joel Marthelot. And I thank you for your attention.
Thank you, Joel, for a great talk. Are there questions?
Hi, beautiful talk. So I had a question about, so you showed us a lot about this folding at the base of the branch. Is this also clear that this would contribute to this sequential folding of the leaves? And then I guess another question in between these two talks is you showed all this beautiful work about the way in which these things unfold. Do you have any sort of design intuition? Is this a particularly efficient way for these things to be packaged, or would there be other ways that you might try to have these sort of large actuations?
Thanks. So we've focused on this large pulvinus because it's larger, and so it's easier to make all the characterization we've done. But yes, the structure of the two other pulvinus are expected to be the same, I would say. So this first question. Second question is, so for example, the way that thing is packed really comes from morphogenesis process just before the adult stage of the, so wing in Drosophila, they have been quite studied, in fact, because they are changing shape regularly in the larval and pupal stage, and they are oscillating between phases in which they are stretched, often by hydraulic pressure. So even in the pupal case, actually, there is a, sorry, in the pupal stage, there is also another phase in which you have stretching of the wing associated with hydraulic actuation inside the wing on folded processes. And so maybe I can show you a movie of this folding process, if I can find quickly. There is a beautiful paper by a Japanese group, Tsuboi, and a co-worker. And so this is so this is in the pupal case, much before the deployment process. And you can see that you start with a flat wing that looks very similar to the wing of the adult. And you have a growth process in which you are connecting the growth process to some patches of adhesion of the wing to the cuticular layer, which at this time is some kind of a bag structure, in which you get this typical folding process. And so I have no idea if this thing is optimal, but it's still a very large variation of shape, and it's a process which is extremely, it's the same in each fly, essentially. It's something which is very controlled. Then it's very different from one insect to another. So there is also a question of, is the folding the same in different species of insects? Especially insects, you have a very large variation of size, actually. So I would say it's an open question. Yeah.
I have a question. So if we talk about like opening and closing of flowers, like which happens at a slower scale, so would that be like, first of all, the first question is, would that be like water driven? And also, like, if I want to describe both processes at a sort of macroscopic scale, would I do it similarly? Like,
yeah, so I don't want to do to say I'm not completely sure what is. So yes, for sure, petals are associated with a variation of flow of water. I'm not completely sure. So maybe Adrien knows better. Actually, you know better. the motion associated with petals in flowers? Okay. So for sure people have looked, but I don't have a clear picture in mind, so I don't want to say. For sure, it's associated with variation of internal pressure, but I don't have a good reference to give you. Yes, so there is a lot of things which are also happening at the base of the structure, right? So some paper have actually shown that there is, you can have some description of the bending which is happening at the base of the petals, which is associated with the large scale variation of the shape. A reference of papers, maybe there is Étienne Couturier that looked at similar question, but in leaves, how leaves are initially folded and unfold during the expansion process of the leaves. Yes.
Would they would they be described differently macroscopically? Like at the tissue level, I suppose they'll be different, but macroscopically, if I want like at a continuum level, let's say if I want to describe it, like a contraction, something like that.
So the petals versus the wings, for example. Yeah, so for example, all the thing I've discussed about the fact that you have stretching in the wings, I think this is completely not the case. Again, I don't want to be, I would be surprised that it's the case in the leaf case. Actually, I think it's completely isometric, the transformation of the leaves.
More questions? Okay. There are no more questions. Let's thank Joel again for a great talk. Just a quick reminder, we have a poster session tomorrow at 11. So there'll be boards put up. You guys can stick your posters on.